Hi, my name is Branden Sanabria, and I'm a second-year PhD student in the Neuroscience program at Rutgers University in the lab of Dr. David Margolis. What is neuroscience, you may ask? Well, neuroscience is the study of how our brain works and how our nervous system works. And in our lab, we're particularly interested in something called sensory motor integration. Sounds complex, right? Well, every day we do things that require us to learn about our environment and make decisions. For example, we learn how to hold a spoon and bring it to our mouth to eat correctly when we eat, or even things like shooting a basketball into a hoop and coloring inside the lines of a picture. All these actions require your brain to detect sensations you're feeling and adjust what you're doing. So, for example, if you're shooting a basketball, it may be that you find that perfect shot and remember how it felt in your arms and your legs, and next time you shoot the ball, your muscles will act a little bit different to help you. Of course, these things take practice and time to learn, and your brain is continually making these adjustments for you. My job as a scientist of the brain is to try to understand how this whole thing is happening and how do we make decisions based on our senses. Well, we can't just open up a human's brain and take them and look at their neurons. So in science, we usually use animal models. And in my lab, we use mice. Mice are very interesting. They primarily use their whiskers to sense their environment around them. So, what I do is I take pictures of the brain cells that are responsible for getting information from the whiskers of the mouse and turning it into a decision for them to move or to not move. And to do that, I use the microscope and I label cells in a bunch of different colors, and it has been a really cool process. Here is an example of some things that we do in my lab. As you can see, neurons come in all shapes and sizes, and we're able to color them in different colors. Using the colors allows us to see what type of brain cell they are, and we can follow their tracks to see where they're connected in the brain. For example, if you look on the left picture, you can see that there's green tracks going all the way to the center of this brain slice. And if we zoomed in even further, we can see it looks pretty messy, and there's lots of green, red, and there's a blue cell in the background. My job is to get really close to these neurons and try to see how these two different parts are connected. Understanding brain connectivity will allow us to cure diseases such as Alzheimer's or movement disorders such as Parkinson's disease and even disorders of learning. I hope you enjoy this and have a good time.